Welcome everybody to the COVID-19 vaccine workshop. We are very, very pleased to present this to you. And today is a very special day. It's the first day. We've got a very exciting program. And this would not have happened without our organizing committee, which I'd like to introduce to you today. And you can see here, we had a fantastic group of people that have helped design this program over the next 2 days. Just a special thanks to Dan Kuritzkes and Jonathan Shapiro, who have been working very closely behind the scenes with me to try and get this program going, and also all the people at Virology Education who are bringing this to you. But thanks so much to the organizing committee. We've got a great program. It's over 2 days. So today, the first session for a couple of hours is going to be on the update of global vaccine situation and our critical unknowns about this. And then the session 2 is about vaccine development. Tomorrow, we're going to talk about something different, what we know about protection, and also we're going to talk about the arrival of the vaccine and how that has affected all of our lives. So we've got some learning objectives to have a better understanding of the pathogenesis of COVID-19 as it relates to vaccines. What constitutes natural immunity? We're going to try and understand the types of vaccines being studied and their rationale, and obtain an updated understanding of which vaccines are in advanced trials and immunological endpoints used there, and hopefully be knowledgeable of the key aspects of vaccine preparedness, manufacture, and distribution. Now, we really value your feedback. We're so thankful you're online today. So please fill out the session survey at the end of each session and participate in the general workshop evaluation, which will come after the event. A certificate of attendance will be sent by email upon completion of this post-workshop survey. Now, some practical tips. We have to do this on all of these webinars because, as you can see at the bottom of here, if you need live support, there is an icon there for technical assistance. But also, really importantly, is to submit your questions through the live Q&A widget, which is usually found at the top right-hand side of the screen. And that's so important for when we have our roundtable discussion, because we love to have and consider your questions that come in from around the world, and it's a really, really valuable thing for all of us on the panels. Also, you can chat and connect through the Connection Hub. So I'd really like to acknowledge now the support, gold level sponsor of Janssen, infectious disease and vaccines from Johnson & Johnson, and our principal supporter of Roche. So thank you so much for your support for this. And we'd like to acknowledge our endorsers of this program, including ARPAC, NATAP, ANOVA, Erasmus, Asham, and Pandora. So I'd now like to hand over to my co-chair, Professor Daniel Kuritzkes, who's Harriet Ryan Albee Professor of Medicine at Harvard Medical School and Chief of the Division of Infectious Disease at Brigham and Women's Hospital in Boston, Massachusetts, the USA. Dan, thank you so much.
Thank you so much, Anton, and welcome to everybody. It's great to have you with us this evening, afternoon, or morning, depending on your time zone. As Anton mentioned, we have a really terrific program, and now the next slide will show you the topics for this first session. We will have an update on the current vaccine situation, an overview provided by Dr. Onyema Ogbuagu from Yale. And then we'll hear about immune responses in current vaccine rollout and clinical trials studies, the impact on durability and transmission from Christiane Eberhardt from the Center for Vaccinology at the University of Geneva. And then our third and final talk will be by Annemarie Wensing from the University Medical Center in Utrecht in the Netherlands, speaking on vaccine responses in key populations with a focus on the elderly, immunocompromised, and persons with HIV. Unfortunately, this afternoon, we won't be having Patricia Brownings join us for the panel discussion, but we'll manage to carry on with the remainder of the panelists. It's now my pleasure to introduce Dr. Ogbuagu. Dr. Ogbuagu received his education in Nigeria. He also worked extensively in Liberia and Rwanda, where he played a major role in training providers of HIV care before moving to the United States and becoming a faculty member in the Yale AIDS program and in their clinical trials program, as well as in the Yale Division of Infectious Diseases, where he is now an associate professor of medicine. Dr. Ogbuagu played a major role in the Pfizer-BioNTech vaccine trial, and it's my great pleasure now to turn this over to him.
First of all, I would like to thank the organizers for inviting me to give a presentation as part of the COVID-19 Vaccine Development and Implementation Workshop 2021 webinar. My talk today is going to focus squarely on an update on current vaccine situation and overview. This will serve as a primer for what should be excellent talks throughout this workshop. My disclosures are listed. My research program has relationships with pharmaceutical groups that are involved in the COVID-19 vaccine development process, none of which would influence my presentation today. For this talk, to provide you the most up-to-date information, I have added data from press releases and preprint servers. However, I have prioritized published data above any of these two. Our outline today will focus on the following. We'll talk about some of the lessons learned from the role of vaccinations in contemporary epidemics. We will discuss the current clinical trials landscapes, lessons learned, and future directions. We'll discuss the vaccination platforms, pros and cons, and I'll give you a 10,000 feet overview of the preclinical studies, safety, immunogenicity, and efficacy data that have emerged from the contemporary clinical trials. And lastly, just a few slides on the vaccine rollout. I wanted to start by highlighting some very important quotes that I have come across that I think convey strong messages to the audience. The first is a comment by an author in the Frontiers of Immunology, highlighting the public health importance of vaccination as being the most successful medical approach to disease prevention and control. And I wanted to also mention a quote by Anthony Fauci, who, when the phase 3 trials were up and running, mentioned that if we get both of these together, an effective vaccine and public health measures, that would really be a game changer for the COVID-19 pandemic. And lastly, a comment by Jonas Salk, a reply when he was questioned on the safety of the polio vaccine he developed. He said, "It is safe, and you can't get safer than safe." So we'll discuss the history of vaccination and epidemics in this slide, but really just wanted to highlight some lessons from the Ebola experience on how vaccines can play a great role in addressing a current epidemic or pandemic. So some of the lessons learned from the recent West African 2014 to 2016 Ebola epidemic that did spread to quite a few countries, even outside the continent, we learned that innovative and accelerated clinical trials that don't compromise safety can be conducted, as has occurred with COVID-19. We've learned that epidemics or pandemics with rapid clinical courses have an advantage. They allow for rapid evaluation of therapies and vaccines, again, as has occurred with this pandemic. We know that it is very critical that global community collaboration by academic institutions, industry, as well as regulators are critical to provide a rapid response to curb some of these epidemics. And lastly, and no less important, is the need to work with local communities and stakeholders who would receive these vaccines to make sure that they're willing to embrace vaccines, especially when they're novel technologies. Now, this is a pandemic vaccine model. On the top part of the figure is the traditional vaccine development model. I think most of us know that the earliest timeline from initiating studying a vaccine to having it approved for use in humans was about 4 years for the mumps vaccine. And these were typically done in sequential stages, preclinical development, clinical development, and licensure, after which production was initiated. But as you can imagine, because of the urgent need to develop a vaccine for COVID-19, there's had to be an accelerated model that has had the ultimate goal of shortening the vaccine development time. And this has included overlapping phases from phase 1 through phase 2 and 3. working with regulators to review data quickly to make decisions on emergency use authorization and approval, as well as initiation of production, having seen an early signal from early phase trials that suggest that a vaccine candidate was promising, along with all the resources that are required to have that done. This model has definitely showed us a quick and efficient way to do things, and I hope it serves us better for current and future infectious diseases for which vaccines are being developed. However, as you can imagine, one limitation to this accelerated process is that you may not have long-term safety data when approval and licensure decisions are made. The vaccine development process has led to key outputs, and I know most of us are very cognizant with the different phases of vaccine development, but permit me to make a few points. I can't highlight how critical it has been for preclinical studies to inform studies in humans, establishing safety, identifying the appropriate and best vaccine candidates to be advanced to human trials. The animal studies have also allowed for viral challenge studies to have that early assessment on if these vaccines can protect against disease. The phase 1 trials have provided very important safety data and have allowed us to calibrate very carefully the doses according to their side effects without sacrificing immunogenicity. The phase 2 studies have also provided critical data on immunogenicity, as well as its performance in patients with different characteristics. And we all know and have celebrated a lot of the success with the phase 3 trials that have provided data on large-scale efficacy and safety. But that's not the end of the story, because we know that as the vaccine is rolled out, post-approval studies and evaluations are ongoing, which are no less important for us to understand, for example, much greater in greater detail, the safety signals for these vaccines. For example, there's a vaccine adverse event reporting system that the CDC and FDA run in the United States that allows us to capture with the vaccination rollout side effects or adverse events as they occur and keep a log of those. Some studies that will be needed will be comparative studies in the future to allow evaluating either different vaccination strategies or comparing vaccine candidates, as well as expanding study protocols to study populations that were left out of the initial phase 3 clinical trial protocols. We're still expecting a lot of important data from the phase 2 and 3 clinical trials to provide information, for example, about the durability of the immune responses and also to continue to access long-term safety signals. We've already talked about the vaccine adverse event reporting system that will provide safety data. And the registries are being established and could be established to continue to assess surveillance for safety events among unique populations such as pregnant women, other immune compromised patients getting vaccines, and may even provide some evidence of efficacy in these subgroups that were not included in the initial clinical trials. We know that there are new variants circulating and new batches of vaccines will be created to address those. Is yet to be seen what the regulatory process would be and how rigorous the expectation would be for these new vaccines. And there will be a talk addressing some of these regulatory issues around the vaccine development process. This is the COVID vaccine clinical trials landscape, which I think many of us are aware of. This is really updated daily by the New York Times. And here you can see really a flurry of activity of multiple clinical trials that are in different phases of development, the minority of which have achieved authorization and a few that have been approved, as well as those have been abandoned for varying reasons. We do know that this is good news because we need as many effective vaccines as possible to meet the global demand and to really have that achieve that elusive goal of herd immunity on a global scale. Towards the right, you have the figure that shows the lead vaccine candidates, their vaccine platforms, which we will discuss in subsequent slides, the phases that are currently ongoing, and their authorization or approval process. This has unmasked the fact that there are variable regulatory standards and approaches in different parts of the world that are not necessarily harmonized. And also to just mention that it's been heartening to see that the lead vaccine candidates, and lead by lead, I mean vaccine candidates that have reported on their phase 3 results have been the newish technologies like messenger RNA technology and viral vectors, which have really opened possibilities for addressing other infectious diseases in the future. This is a cartoon of the vaccine approaches for SARS-CoV-2 virus, and this is an image from the frontiers of immunology. This just goes to show that multiple approaches are being evaluated for vaccines for SARS-CoV-2. In brief, there are nucleic acid-based or gene-based strategies, which can be either mRNA or DNA vaccines. There are viral vector technologies, which include both non-replicating viral vectors and replicating forms. There are whole virus vaccines that can either be live attenuated or inactivated or viruses, protein and peptide subunit viruses, nanoparticle and virus-like particle vaccines, as well as immune cell therapy to address the virus. However, we do know as these approaches are evaluated in clinical trials, the key considerations would be how immunogenic they are, how safe, effective, what kind of delivery systems there are, the dosing requirements, storage requirements, and amenability to mass production, which is critical for the raging pandemic. Here are some, not an exhaustive list of the lead vaccine platforms, but I just wanted to utilize this opportunity to highlight some of the pros and cons of each approach. Towards the left, we have a messenger RNA, which we know is a potent stimulator of the immune system, easy to produce, mass produce, which is an advantage in this setting. However, some of the limitations include, because of the fragile nature of the messenger RNA, that it needs a cold chain, and also the lipid nanoparticle formulation, which forms that protective shell for the messenger RNA, can be one of the reasons why some allergic reactions are occurring. For the viral vector vaccines, they have the advantage of really stimulating robust T-cell responses. However, one limitation are anti-vector immunity. For example, the adenoviruses could face limitations with high anti-vector immunity in African populations, for example. Inactivated viruses tend to be safer than using live attenuated, and you can imagine that's a desirable property for vaccines that you'd wish to use in people with compromised immune systems. However, the production process is quite laborious. Protein subunit vaccines, we have a lot of experience with. They're relatively safe, they're cheap to produce, but they tend to trigger less T-cell responses and require adjuvants for optimal immunogenicity. It's been nice to see that there's been tight coordination between the clinical trial groups as well as regulators like the FDA and agencies within the World Health Organization. And these were critical early on to inform the design, desired outcomes, and approval requirements by these agencies. To the left are some of the guidance around high quality studies that could be conducted. Typically, these would be placebo-controlled, randomized controlled trials, representative of different racial ethnic groups, representative of the populations within a setting that have the disease, with a good sample size to be able to show significant outcomes. Also, with the requirement for a decent duration of follow-up, 2 years, so to adjudicate longer-term safety outcomes as well as durability of vaccines, and importantly, to show efficacy against preventing COVID-19, including severe disease, given some of the concerns of antibody-mediated, antibody-enhanced disease. Similarly, built into the protocols were robust interim assessments for safety boards to assess for efficacy or futility of the intervention, as well as to detect any safety concerns during the conduct of the study. To the right are some of the requirements for emergency authorization or approval, including significantly that satisfactory manufacturing, quality controls, and facilities have to be in place. A desire that vaccines should have greater than 50% efficacy, sufficient, at least minimum 2 months safety data post last dose of vaccine. And the World Health, in addition, had set out prioritization characteristics, including that single-dose vaccines would be preferred over double-dose vaccines, and that also having vaccines that produce at least 6 months durability of protection would be desired characteristics. Now let's talk about the different phases of studies and what we've learned to date. Preclinical studies have been critical and instructive and to inform subsequent human phase trials. For example, studies in mice and non-human primates has allowed evaluation of vaccine antigen constructs to select the best candidates. They've allowed for immunogenicity studies as well as immune profiling, which have been critical to assess those parameters in animal studies, and also have allowed for viral challenge studies that have allowed us to answer important questions, like if it prevents asymptomatic disease or replicating virus. And these have really been critical to inform human trials and provide some very valuable data. Let's talk about safety. So safety assessments have been built in strongly into the vaccine clinical trials protocol. To the right is a figure from the Moderna study, looking at the messenger RNA vaccine approach. We know that for the clinical trials, safety monitoring was built into the protocols so that there were interim reviews to assess for harms that would lead to either triggering a pause or halting clinical trials. And we saw that kick in for a couple of vaccine candidates where there was events that had to be reviewed closely before the trials were allowed to continue. And that should really reassure individuals that safety was a key goal or target for these studies. The studies typically included actively collected data on solicited side effects, which were intensively collected for the first 7 days or so post-vaccination, and also unsolicited adverse events were also captured. Now, the figure on the right shows that after the first vaccination, that individuals could have anything from local reactions at the site of injection, which can be pain, redness, or swelling, and also systemic reactions that could be flu-like symptoms, fevers, chills, fatigue, and arthralgias. Looking at the figure on the right, comparing reported side effects from vaccination 2, you can clearly see that after the second vaccination, that there was more frequency of these events and also increased spectrum of severity. However, the good news is that the majority of these side effects are mild to moderate. The commonest local reaction is injection site pain, and the commonest systemic reactions are flu-like illnesses. The good thing is they occur early, usually within a day or two of injection, and resolve pretty quickly. We found that older individuals tend to have less reactogenicity in these trials. The good news is that there is a very low incidence, generally, of severe adverse events, particularly vaccine-related adverse events supporting decent safety profiles. Let's talk about immunogenicity. Immunogenicity assessments have been critical to assess both humoral and cell-mediated immune responses to vaccination. To date, they have focused on antibodies, the spike protein, which is the vaccine antigen, for the vast majority of the vaccine approaches, other than whole viral approaches, or targeting the receptor binding domain. They've also assessed neutralizing antibody titers as well as cell-mediated immunity. If you look at the figures on the right, this is a dose-ranging study for the Pfizer mRNA study that evaluated 10 micrograms, 20 micrograms, and 30 micrograms study. Everyone received 2 doses of vaccine at day 1 and 3 weeks later at day 21. The top 2 boxes represent the antibody levels, IgG, in younger individuals to the left, age 18 to 55, and older individuals 65 to 85 to the right. And the lower boxes, it represents the 50% neutralizing titers, again, in younger individuals to the left, older people to the right. Now, immunogenicity studies have used convalescent sera from healthy individuals as the comparator or the bar for what the target antibody levels and neutralizing titers should be. And this is depicted by the yellow horizontal line on both parts of the figure. So many of the platforms have shown that after the first dose, you don't really achieve that target concentration. However, the second dose acts as a boost, and that's where individuals across the board tend to approach or exceed the target antibody or neutralizing titer levels. So this have shown the importance of second doses as a boost for majority of the vaccine platforms. And longitudinal studies remain ongoing to assess the levels over time. However, an important thing to mention is that the correlates of immunity are still being worked out, and hopefully with phase 3 trials, looking at individuals, for example, whom had breakthrough infections with vaccination and what their antibody levels were, we may learn more about the correlates of immunity. Important to mention again, we saw that older individuals have lower reactogenicity, and similarly, they also have lower neutralizing antibodies across the board. So to the figure to the left shows different vaccine approaches, viral vector approaches, inactivated viral vector approaches, and messenger RNA. And you can see in all these vaccines, comparing younger people to older individuals, there's a decreased neutralizing titer for all these vaccine technologies. However, the figure to the top right shows you reassuringly that in spite of the decreased neutralizing titers in older individuals, they remain above and exceed that of convalescent sera titers. And we have seen that, that this is clinically important because in the phase 3 trials, it appears that in spite of what appears to be lower antibody levels or neutralizing titers, it doesn't appear that age is impacting efficacy. Another caveat is that the level of neutralizing titers or antibodies do not always correlate with the degree of immunity against the disease, and there are many immune responses that protect against viral infections that we don't measure with either of these approaches. A question that's frequently asked is what's the durability of the vaccine responses? It remains a work in progress. However, data continues to emerge from the earlier phase trials about the durability of responses following vaccination. This is data from the Moderna messenger RNA study that evaluated trends of antibody levels to the receptor binding domain or pseudovirus neutralization assays over a 120-day period. Now, they found that the binding and neutralizing geometric mean titers exceeded those of convalescent sera, and this was convalescent sera obtained from individuals who are at least a median of 30 days or about a month from the time of their COVID diagnosis. As these studies continue, we will continue to learn more about the durability of protection. And for now, it looks good at 4 months post first vaccination. Let's discuss efficacy. There's been a plethora of reports, especially from the phase 3 trials on the efficacy of vaccine candidates. In this table, we show the efficacy reports to date, again, favoring published data. The messenger RNA technologies with Moderna reporting a 94% efficacy against symptomatic COVID-19 and Pfizer reporting 95% with the same endpoint. We saw different results from the AstraZeneca study, depending on if you got a standard dose first and a standard dose later, or half dose first and a standard dose later, averaging about 70% efficacy. We saw and heard recently about from the viral vector study, Johnson & Johnson single dose vaccine, about a 66% overall efficacy, as well as from Novavax about their efficacy showing differences by variant. It's clear from studies that were conducted in parts of the world that at the time where there were circulating variants, for example, the variant that was identified in South Africa with the concerning 484 mutation in the spike protein genome, we see that there was, it took a hit on efficacy, both for the Johnson & Johnson vaccine as well as the Novavax vaccine. Good enough is that many of these trials, beyond having met their primary endpoint, are expanding their protocols to groups that were previously left out of initial trials, including a little bit of younger children and adolescents and individuals with certain immune compromising conditions. And we hope and in the future expect that we would have even more data in groups like pregnant women and even younger children. Important to mention that vaccines also have shown protection against asymptomatic infection, which is important from a transmission standpoint. I decided to share this data, which is a preprint from the Lancet regarding the AstraZeneca vaccine. And here they looked at the vaccine efficacy as it relates to the time of administration of the first dose of vaccine. And so in this study, they conducted surveillance for asymptomatic infections by weekly swabs of the nose and throat. So overall, and the bottom line results are that any PCR positive cases of asymptomatic infection were reduced by 67% in individuals even after a single dose of vaccine, suggesting a 2/3 reduction in PCR positive nasal swabs anytime from 22 to 90 days after the first vaccine. A phase 3 study of the Moderna vaccine also showed, even though the numbers were low, 67% reduction in asymptomatic infection as well. Now, this is a model I have created that shows what appears to be the relationship between protection against disease severity and vaccine efficacy. And I think if you look at the most recent or contemporary phase 3 clinical trial reports, it appears there's a graded increase in protection against disease based on disease severity, so that the point estimate for protection against asymptomatic infection tends to be on the lower end, and while you have excellent protection against severe disease and death. However, the protection against moderate to severe disease appears to be influenced by certain things, like the presence or absence of circulating variants that can evade those vaccine-generated immune responses, host characteristics like being immune compromised, and we also need to pay attention to the endpoint definition in clinical trials. However, the finding that the vaccines all have excellent efficacy against severe disease and death are really heartwarming from a public health standpoint. This is a slide that shows the protection that occurred over time in individuals who got at least, these are the 2-dose vaccines. So many of these studies, be it the AstraZeneca vaccine or the Pfizer vaccine, the Sputnik adenoviral vector vaccine or Moderna and mRNA study, these studies have always showed, these are the Kaplan-Meier curves of cumulative incidence of COVID-19 in individuals who received placebo and compared to those who got vaccine. And if you look across all the figures presented here, you can see a separation and happening in many instances after the receipt of the first dose and before the second dose, that you already see an efficacy signal in these vaccines. And so we do know that even the first doses of a planned 2-dose series vaccine can confer significant protection against COVID-19. But however, you can clearly see that you need the second dose, and after the second dose, that's typically when the peak efficacy is achieved. There could be a time component to it after receiving the first dose, as we see with the single-dose Johnson & Johnson vaccine, for example, where the protection was measured at about a month after receiving the first dose. Now, the good news is that in general, it appears that certain patient characteristics like age, race, ethnicity, obesity, presence of comorbidities, and prior SARS-CoV-2 infection do not appear to be impacting efficacy, but we will see if these hold on longer-term follow-up. Important to mention clinical trial populations that were included and excluded. We know that for now, most studies have occurred in healthy or non-immune compromised adults. However, clinical trial protocols have been expanded, including the Pfizer study, for example, to HIV-infected patients with well-controlled disease. And also, clinical trial protocols are expanding to even younger children, age 12 and above. However, there remain significant data gaps in kids younger than age 12, pregnant and lactating women, and many categories of immune-compromised patients. So to wrap up the vaccine and clinical trials presentation, just need to remind you that vaccine efficacy appears to improve across the spectrum of disease with the greatest efficacy for preventing severe disease and death. The first dose can provide decent efficacy that exceeds that predetermined 50% threshold, but that the second dose in many of these vaccine platforms boosts immunogenicity and may contribute to durability. We've seen that elderly individuals exhibit lower immunogenicity and reactogenicity, but vaccine protection against COVID-19 appears unaffected. And we know that because we have many effective vaccine candidates available, the era of placebo-controlled vaccine trials, which would allow other vaccine candidates in earlier phases of development to proceed, appears to be closing, if not closed already. Just a few thoughts about vaccine rollout, herd immunity, and challenges to wrap up. Just to mention that achieving herd immunity is usually a composite of the seroprevalence in the community and in addition, vaccine-induced immunity, so that where the seroprevalence is high, you do not need to have that much vaccine coverage. So for example, the estimate is that the UK and US have about a 5 to 20% seroprevalence of COVID-19. And so in that case, you really need just about 50% of the population to receive vaccines to be covered. So we all need to work with these kind of models to help us determine what the optimal vaccine coverage should be. We do know that it is more desirable to get vaccine-mediated immunity than natural immunity because it's more durable. We know that strain-specific immunity would favor vaccines because immunity from prior circulating strains may not hold up against a new variant. And we know that the number of vaccinations needed for vaccines that are less effective would need more. This is another model that assesses or produces evidence on the impact of triaging vaccinations to different groups on transmission and mortality. In this model, they looked at vaccination strategies involving younger individuals, older individuals above the age of 60 in the community, or distributed across all ages. And these are models that are important because of vaccine shortages as they exist. You can model based on the percentage of vaccine supply, the effects on infections and cumulative mortality, and also work out the reductions in infections and deaths over time. So for example, in this model, vaccinating older individuals had the greatest impact on cumulative mortality over time. However, vaccinating younger individuals had the greatest impact on reduction of infections, as you can see here. So these are critical, and scenarios vary between countries and within countries. And so, models would be really important to inform rollout. Another approach is a risk-based approach, and this is taken from the CDC website that looked at the data for some underlying the comorbidities listed as conferring an increased risk of severe COVID, with the most convincing evidence being those conditions on the left, like cancer and pregnancy, and conditions which could confer increased risk, but for which there's limited evidence, like HIV and liver disease. Having these data are critical to inform prioritization of risk groups for receiving vaccinations. We know that vaccines are currently rolled out globally. As of yesterday, there have been about 168 million doses administered, with an average of almost 6 million doses a day. To the right, you can see the countries listed by their income category, whom have both either confirmed purchases of vaccines or entered agreements to purchase vaccines. In many of these cases, some countries have actually purchased more vaccine than they need for the entire population together. And we can clearly start to see the malavailability or maldistribution of vaccines by income category. Many low middle income countries are yet to start vaccination, unfortunately. In addition to that, logistics for vaccine administration and vaccine hesitancy remain key barriers to optimizing the real-world effectiveness of COVID vaccines, and these will be further elaborated in talks to come. I just wanted to highlight some of the countries that have been top performers and really have been informing real-world outcomes for vaccination strategies. The last slide I will show are some of the approaches to vaccine strategies that can be informed by clinical trials. For example, extending duration of vaccination interval that allows for production of vaccine and administering vaccines to catch up with the demand. To the right is the data from the AstraZeneca study showing that as you delayed the duration, the interval of administering the two-dose strategy, that we actually found an increase in efficacy. The caveat here is that this has only been shown for viral vector vaccines, and we do not have this data for many other vaccine strategies. There are also mix and match approaches that allow you to get one vaccine and get another type subsequently. These are only being conducted as clinical trials. One has been launched in the UK, as is shown below. So let me wrap up this presentation by saying that we have seen that there are multiple phase 3 trials that have shown variable but robust efficacy for many vaccine candidates, especially for the desired outcome of preventing severe disease and death. For most of these strategies, 2 doses are required for optimal efficacy and possibly durability. Safety records have been great. We see that emerging variants do pose a threat, both current and what would be future if the pandemic continues and booster doses are being prevented. And we cannot overemphasize the role of modeling to inform vaccine rollout prioritization, and hopefully we can address equity with vaccine distribution globally to get to the end of the pandemic. Thank you again for having me.
Thank you so much, Dr. Ogbuagu, for your fantastic overview and plenary talk on the current situation with vaccines. And during the next couple of days, we're going to dig deeper into each of these topics and hopefully present to you quite a very deep overview of what's going on. It's a great pleasure for me to present the next speaker, who is Christiane Eberhardt. Christiane is a physician scientist with a background in vaccine immunology and is currently working as clinical vaccinologist at the Center for Vaccinology in Geneva. She's worked under Professor Claire-Anne Siegrist and Professor Rafi Ahmed on human B-cell and CD8 T-cell immunology in the context of vaccination and viral infections. And she's also conducted a policy-changing clinical study on the timing of maternal pertussis immunization, a work that was awarded with several prizes. So we're very honored to have you speak today about immune responses in current vaccine rollout and clinical trials, and something that's really concerning us all, the impact on the durability and any transmission. Dr. Eberhardt, the scene is yours. Thank you.
Good afternoon, good evening, everybody. It's a pleasure to be with you today. Thank you for the invitation, and I have nothing to disclose. Today, we are talking about the understanding of the principles of immune responses to vaccination. We will look into the assessment of immune responses to COVID-19 vaccines, especially measurement of humoral and T-cell responses, the durability of vaccine responses, and a potential response to a boosting dose. We look into the impact on transmission, and we will end with consideration of immunological data as potential correlates of protection, because as you know, there is no correlate of protection yet for COVID-19. Let's get started with the understanding of the principles of immune responses to vaccination. What happens after vaccination? We have the immediate innate immune response. What is that? So you get the vaccine, and then you will induce monocytes, macrophages, natural killer cells, and also dendritic cells. They will do two things. First, they will create an inflammatory environment. And second, they will pave the way for the adaptive immune response, which means they will present antigen to B and T cells, and they will activate them, and thus they can induce their adaptive immune response. Often we observe local reactions such as pain or redness at the injection site, or systemic effects such as fever. And these partly reflect the activation of the innate immunity. There are some markers of innate immunity that can be measured in phase 1 or 2 trials. So for example, we can look at the blood level or the phenotype of monocytes and NK cells. We can look in peripheral mononuclear cells at gene expression patterns, or also we can measure plasma cytokines and inflammation markers such as CRP. And all this takes place very early, so immediate after vaccination, but during the first days after vaccination. And then following this response, you see from day 4 onwards and up to 15 or more days, we have now the adaptive immune response. So the adaptive immune response, we can distinguish 2 pathways. One pathway is the B cell activation. And as you can see here, it's in the lymph node where you have B cells that are activated and then either develop into antibody secreting cells, which are also called plasma cells, or into memory B cells. And these memory B cells, they are the cells that circulate in the body. They do not secrete antibodies, but they can be activated when you get exposed to the infection or to another vaccination, and then they start producing antibodies. And these antibodies can be induced by all vaccine types, which is important, and you will see further down the presentation. And the neutralizing antibodies, these are the antibodies that they can limit viral invasion. And the other pathway is the development of a T cell response, and it's CD4 T cells and CD8 T cells. This is a little bit more complex, and we are going to spend some time on this topic. So let's take a closer look. T cells recognize very small linear peptides as opposed to antibodies that can recognize whole proteins. So what happens when a cell gets infected or in an antigen presenting cell is that you have the ingestion of the whole protein. This protein gets degraded and it becomes peptides. So it's 8 to 10 or 15 amino acid long. And these peptides are presented on molecules that are called MHC or HLA molecules on the surface of either the infected cell or of an APC. And these are recognized either by CD4 T cells in case of antigen-presenting cells, or by CD8 T cells on the surface of infected cells. And as you can see, because it's not the whole protein, and if we talk about the new variants that appear during this COVID pandemic, you can appreciate that it's only a small peptide. So if there is point mutations and very small changes to the S protein, for example, for COVID, the impact on a T-cell response will be much lower than compared to, for example, neutralizing antibodies. And what do CD4 and CD8 T cells do? So CD4 T cells, they can be induced by all type of vaccines, so by live and subunit vaccines, and they produce various cytokines, and they are the ones that are essential to help building antibody responses, and you might know them under the name of T helper cells. The CD8 T cells, these are the killers. They are mainly induced by live vaccines, and we think that they are possibly also induced by nucleic acid vaccines. As the name says, they can kill virus-infected cells, and their other important role is to produce interferon gamma, an inflammatory cytokine, which contributes to the termination of infection. With all this knowledge now in mind, we will have a look into the measurements of humoral and T-cell responses that had been undertaken in the COVID-19 vaccine trials. But first, let's define a little bit better the antibody responses. So we distinguish between different types of antibodies. We can quantify the binding of antibodies, which means it's the capacity to induce an immune response, but it's no direct indication of protection. And then we can look into the quality of antibodies. So it's the neutralizing antibodies, which suggest a potential protection because they can neutralize the virus and thus inhibit invasion. But the protective level is unknown before phase 3 trials. And in the case of COVID, as you know, we don't know yet the protective level either. And there is other functional activities of antibodies, which is, for example, the affinity of antibodies, which means how well they can bind to the antigen, and also the so-called Fc binding capacity, which contributes to the antibody-dependent cell toxicity, also known as ADCC. So if you look at the antibody and neutralizing antibodies in the COVID trials, just a little reminder. So here you can see an example of the SARS-CoV-2 virus. And on the surface, as you all know, there is the so-called spike protein S, which contains the receptor binding domain. And it's the receptor binding domain that binds to ACE2 and thus helps binding to the cell and invading a cell. And so if we measure antibodies that are directed against RBD or the spike S protein, we talk about binding assays, and we do this by ELISA, and we can quantify antibodies. But if you want to look at the functional assays, then we have to do so-called virus neutralization assays to see if the antibodies that are in the blood of patients can really neutralize the virus and inhibit cell infection. So if you look at one example, it's, for example, the Moderna vaccine trial. You see antibody titers or neutralization titers. And if we concentrate on the upper panels, you see it's the anti-S protein antibodies and the anti-RBD protein antibodies that were detected before vaccination, and then before the second dose of vaccination that was given after 4 months, and then it was followed up until day 60 around. And you can see that there's an increase in anti-S antibodies, and there's also an increase in anti-RBD antibodies, and this is true for the dose that is currently given in the vaccine, as well as in the lower or in a higher dose. And then we're interested, as I said, in the quality of antibodies, and we can do this by different type of neutralizations. There is a so-called pseudotype virus neutralization assay, which is a recombinant virus that is used to mimic the infection process. And because it's not a SARS-CoV-2 virus, this needs a lower level of biosafety and can be done in more in less specialized labs. And then we have the live wild-type SARS-CoV-2 neutralization, where the real virus is used, and this requires a high level of biosafety, a BSL-3 lab, for example. And here again, you can see that there is a slight increase after the first dose of neutralizing antibodies, but it's less pronounced as it's here if you just look at the binding capacity. And then we reach again a plateau after the second dose. And as you can appreciate, there's way fewer patients that have been tested here, because this is a very labor-intensive and a very specialized test to be done. And I want to draw your attention also on the last part of each graph, which is the convalescent graph and the convalescent serum results. And why is this necessary? As I told you, we do not know the correlate of protection. So we do not know for each assay how much antibodies are necessary to confer protection in the patient. And because every lab is doing a different test, we cannot compare in between vaccine trials. So it is important that we standardize to something. And in this case, we standardize to convalescent sera. But I can hear you already say, well, convalescent is not convalescent, because some patients had mild disease and some patients had severe disease and were on intensive care. And you're right. So if you see such a graph, always check the disease severity that was included, if it was only very mild patients or if it was really severe patients where we expect a higher antibody response. And also another finding I just want to share with you is this is another vaccine trial. It's the AstraZeneca Oxford vaccine trial. And here you see 4 different types of neutralization assays in the same patients in the same trial. And I just want to show you that if you just look at the numbers, right, this leads to errors, because the number and the titer for each assay is very different. So have a good look when you analyze these data and don't compare it in between assays, not even in between the same patients. Now let's look at the measurement of T-cell responses. And I would like to share with you a method that has been used in many of these trials, and just to help you understand what it means if you see such a graph, which is here on the right, with spot-forming cells per million PBMCs, which can be a very abstract concept. So how is this functional T-cell assay, also called ELISpot, done? What we do is we use peripheral blood cells, which contains T-cells, and we add to those cells SARS-CoV-2 antigens, for example, the S protein, and we put this in a 96-well plate. And then bottom of the 96-well plate, there is a filter kind of paper, which has anti-interferon gamma antibodies. And then we put this in an incubator overnight, and what happens? It happens that the T cells that are specific for SARS-CoV-2 or for the antigen that you added, they are going to be activated and they're going to produce interferon gamma. And this interferon gamma is going to be bound to the antibodies on your filter paper. And then you can develop it, and then you count spots. And each spot corresponds to an antigen-specific cell that has produced interferon gamma. One little thing I would like to share is I told you that CD4 T cells and CD8 T cells are capable of producing interferon gamma. And this is a preprint from the Pfizer vaccine trial, where you can see that they isolated CD4 T cells and CD8 T cells and only put those onto the ELISpot. And you see that, for example, for RBD, it's mostly CD4 T cells that recognized RBD. But for the S protein, it's both CD4 and CD8 T cells. So if you look into those graphs, which is from the AstraZeneca vaccine trial, where you see spot-forming cells that produce interferon gamma, you know that you don't look only at CD4 or CD8 T cells, except if it's specified, but you usually look into PBMCs, so both CD4 and CD8 T cell responses. And again, be careful. We do not know how many spot-forming cells are necessary to confer protection or to participate in protection. So the CD4 T cells, how can you better qualify those? we have different T cell subsets of CD4 cells. And the main ones are the Th1, so the T helper 1 cells, which are responsible for pro-inflammatory responses. And for example, they produce interferon gamma, as I showed you before in the ELISpot assay. Then we have Th2 cells that are more involved into allergies. They produce other cytokines, and they are less efficient in ending an infection, for example, and by a viral infection. We have Th17 cells that play a role in inflammation and mucosal defense, and you see they have other cytokines they produce. And then we have follicular T helper cells, which are localized in lymph nodes, for example, and those are the ones who help B cells to differentiate into antibody producing cells. And how can you identify those? Well, you do this by flow cytometry, and you see in the Moderna vaccine trial that you look into interferon gamma, IL-2, and TNF alpha production, and you see that after the first dose, you have a slight increase in those Th1 cells, and then after the second dose, you see a booster effect where you have a higher amount of those Th1 CD4 T cells. And then for the CD8 T cells, I told you that they are the killer cells, so they control infection and clear intracellular viruses by a direct killing of infected cells. So what do? Is they are in contact with the infected cells. They release perforin, which kind of makes a hole in the cell, and then they put granzyme into a cell, which induces apoptosis. And also they're involved in the indirect killing of infected cells because they release cytokines, especially interferon gamma, which then induces an inflammatory environment and helps clearing the infection. And here is an example of flow cytometry. What you can see is this is before the vaccination and after the vaccination. And this blob here is CD8 T cells. And then if you see cells here in this part of the chart, they produce interferon gamma. And if you see them here, they produce IL-2. And if you see them here, they produce both IL-2 and interferon gamma. And you see that after the vaccination, they are cells, CD8 T cells, that produce interferon gamma. And those cells are specific for SARS-CoV-2. And then this translates in a graph, for example, in the Moderna trial, in these results, where you can see a slight increase of CD8 T cells that produce interferon gamma, IL-2 or TNF after the second dose. But again, we do not know how many of those CD8 T cells are necessary to, for example, have less symptoms or to faster clear the infection. Now, what do we know about the durability of vaccine responses and responses to a boosting dose? Well, we do not know a lot. This is pretty quick. So on the left side, you see a binding assay, the anti-RBD antibodies in the Moderna vaccine trial. And it was recently published where you can see the increase after the first and second dose. And you kind of see a plateau until day 120 after vaccination. And this is in the younger population. And if you look in the little bit older population, you see a slight decrease in the binding assays. And then if we look at the wild type neutralization assays, you can see an increase and then a slight decrease in younger patients and a little bit more pronounced decrease in the patients that are a little bit older. But again, one line is one patient. So we talk about 9 or 10 patients in total, which is very hard to draw conclusions to the whole vaccinated population. So if we do not know so much about durability, and if we see that antibody titers decrease, we are now obviously interested in knowing if these vaccines have induced a memory response and that we can give a booster dose and we can recall this memory. And I just want to explain to you, obviously there's no data yet on any recall response after this vaccination, but I want to show you the principle so that once these data come out, you can better appreciate the results. So if you give a vaccine boost, what happens? I showed you that there is memory B cells that circulate and do not produce antibodies. But if they encounter the antigen, they can either directly develop into antibody secreting cells, or they can undergo, again, the same kind of interactions with the T cells in the lymph node, and they can be more and more specific and produce more and more memory B cells. And this is another way to show you, if you look at the antibody titers, after the first dose, we have a slight increase. And then after some weeks or months, if we give a booster dose, then we have an increase that is much faster because you already have these memory B cells and they do not need 2 weeks to develop and to produce antibodies, but they can do it in some days. And we can reach higher titers and also a better quality, because as I showed you, they can undergo again the maturation process in the lymph node. So these pre-existing memory B cells that come from a former infection or a vaccination, they have an impact on the magnitude and the specificity of the immune response to vaccination. And what about the impact on transmission? What do we know? So we do not know a lot either, as you know, but I want to show you some data from non-human primates, because we can do things with them that we can't do with humans. We can look at the mucosal immune responses, because this is where the virus sits and might replicate. And in bronchoalveolar lavage, they looked into IgG and IgA antibodies. And you see that after vaccination, there is a high antibody titers for IgG, a little bit lower for IgA. And if you look into the viral load there, you can see that in the bronchoalveolar lavage, you can decrease the viral load. But it's interesting to see that on the nasal swab, you still can have COVID or SARS-CoV-2 virus, and you can still detect RNA. If we look into the SARS-CoV-2 vaccine trial that had been done, they swapped patients before the first dose and before the second dose, and it's a low N, but you see that in the vaccine group, there were 14 that had asymptomatic infection at the time of the second dose, whereas 38 in the placebo group, which might suggest that there is a decrease in asymptomatic infection, which might give us a hint on the impact on transmission, but we will need real-world data to better draw any conclusions. And let's finish with considerations of immunological data as correlate of protection. What is a correlate of protection? It is a marker of immune function that statistically correlate with protection after vaccination. And there is two types. There's mechanistic correlates of protection. So it's a mechanistic cause of protection, which means, for example, hepatitis B antibody titers, they protect from disease because they are going to neutralize the hepatitis B virus. And there is non-mechanistic correlates of protection that do not cause protection, but they can predict protection because they correlate with other immune responses that are protective. And this is especially important for mucosal responses that we cannot measure in humans, but we can, for example, measure serum IgA antibodies in the case of rotavirus vaccines. And sometimes we need to do a complex integration of multiple data. For example, this was shown for Ebola, to have a correlate of protection. And here you can see that for Ebola, they use preclinical data on viral challenges in monkeys. They use data from survivors, so correlates of survival, and from clinical trial. And all this led to correlates of protection induced by Ebola vaccines. So today you saw that vaccination induces innate and adaptive immune responses. You saw that the assessment of COVID-19 immunogenicity comprises measurement of B-cell responses and their functional neutralizing capacity, measurement of relevant T-cell subset responses, and the integration of collected data that may lead to the identification of correlates of protection that we do not have yet for COVID-19. We looked into the durability of COVID-19 vaccines, and we do not have sufficient data yet, but I showed you that we can reactivate immunological memory by vaccine boosting. And you looked at the impact on transmission, which is also an open question and for which we will have more data hopefully soon. With this, I would like to end. Thank you very much for having me, and happy to answer questions during the question and answer session. Thank you.
Thank you so much, Dr. Eberhardt. That was really a superb review of the immunology of viral infections and the kinds of immune responses we need to induce with vaccines for protection against diseases like COVID-19. It's now my great pleasure to introduce our next speaker, Annemarie Wensing, who will be talking to us about vaccine responses in key populations, including pregnant women, the elderly, and immunocompromised individuals, such as those living with HIV. Many of the questions that have been coming into the chat pertain to this topic, so I think you'll find this to be a very informative talk. But Dr. Wensing is a clinical virologist at the University Medical Center in Utrecht and an honorary professor at Wits in Johannesburg. She obtained her medical degree from the University of Utrecht and also trained as a clinical virologist, where she received her PhD on the transmission of drug-resistant virus. Dr. Wensing has been a long-term colleague and friend since we both work in the areas of HIV drug resistance, and her important work on HIV cure, particularly as one of the leaders of the EC STEM program to look at HIV cure by stem cell transplantation. And she's been a member of the WHO drug resistance ResNet panel and the IAS USA resistance mutations panel. And like so many of us in the past year, has pivoted directly to the study of COVID-19. So without further introduction, Dr. Wensing.
Good evening. I was asked to talk about vulnerable population and COVID vaccination. And of course, in my position, the first group that you think of are people living with HIV. And if you look at the data, there's actually only anecdotal evidence showing that the serological response to SARS-CoV-2 in well-controlled people living with HIV, so people with an undetectable viral load, is actually similar to that of non-HIV-infected patients. These are very limited data series. If we go to clinical trials with COVID vaccines, there's actually some information about HIV. The FIDSA vaccine had 120 well-controlled people living with HIV included, but they were not included in the primary efficacy and safety analysis. And their data will be analyzed later. If we go to the other mRNA Moderna vaccine, then there are 176 people living with HIV, but none of them, luckily, developed symptomatic COVID-19, giving us limited information then on this particular group. If we look at the AstraZeneca Oxford vaccine, and this is the South African trial, COV-005, and there is a specific HIV cohort, but it's not yet analyzed. Finally, the Janssen adenovirus vaccine has actually 2.8% of participants that are people living with HIV, but they have not been separately analyzed. So this population is present in the trials. We do not have specific information. Some of them have been actually included in the overall analysis. So if there would be a severe alert signal, we would have received it. So generally now, we do consider the vaccine safe for people who have well-controlled HIV infection. We have not particular specific information. We then look at another vulnerable population that has received some attention. Then we're talking about the elderly. And generally, elderly tend to have poor responses to vaccinations. And they have reduced adaptive immune response and different regulations of cytokine production. And the immunosystems render older adults more susceptible to infection and hinders the development of protective immunity after immunization quite often. Also, antibody responses may be lower, but this is not always the best correlates for infection, for protection of infection. For instance, influenza trials have learned to us that even with lower antibody responses, T-cell mediated immunity evoked by the vaccine may be sufficient to protect people. So what have we learned about COVID in general in this adult elderly population? Well, people with age above 65 have a 2.6 higher likelihood of having hospitalizations due to COVID-19 in the US setting compared to people who have ages from 18 to 44. As of December 2020, the cumulative COVID-19 associated hospitalization rate was 1.211 per 100,000 for people over 75 years, and much lower, almost half, for people who have ages from 65 to 74. Also, the risk for COVID-19 associated hospitalization doesn't only increase with age, but also with the number of underlying medical conditions. And of course, people with higher age have often more underlying medical conditions. But we don't have to panic. There is quite some data now about the vaccination about this elderly population. If we first look at the Pfizer vaccine, the vaccine that was actually approved first, It's a vaccine that's currently given in two doses, the two doses. And if you look at the phase 2/3 trial that's still ongoing, there's an immense amount of people included, more than 40,000. They're randomized 1-to-1 to placebo, and actually quite a large group, more than 42% is over 55 years. And in this trial, the primary endpoints were efficacy of the vaccine against laboratory-confirmed COVID-19 infection and safety. And then two-dose regimen conferred 95 protection, as we heard probably before today, in adults. And here you see in this table, you see the different age groups. And what you can see is that actually the numbers are lower with people of higher age. The vaccine efficacy actually stays quite high, but you have to see that in the older age group from 65, especially above 75, confidence intervals tend to be larger. But still, the efficacy is, and the anecdotal evidence in the smaller group looks really, really good. What looks also very good is the side effect profile. Here you see the local events, and here you see the systemic events. And these are the people who are 60 to 55, and these are the people who are older. And you can see there's a trend to lesser side effects in those who are older, which is actually quite nice. If we go to a similar vaccine, also mRNA vaccine from Moderna, also here 2 doses confirmed a really high protection rate against COVID-19. In phase 3, there are over 30,000 people and double-blind randomized 1-to-1 to placebo. And here you see the impressive protection from the mRNA vaccine compared to placebo. The primary endpoint here was the efficacy in preventing symptomatic COVID-19 with onset at least 40 days after the second injection in the per-protocol population. And was very hopeful that efficacy was similar across participants who were elderly, above 65 years. And here you can see, again, vaccine efficacy in this older population is almost similar to the younger population, but again, the confidence interval widens here, especially also because the number is lower of people with higher age in the trial. If we look at specific subgroups in this trial, then they have also looked at people who have severe risk factors for COVID-19, for instance, with chronic lung disease, cardiac diseases, severe obesity, liver disease, or people living with HIV. This subgroup are only people below 65, probably because of low numbers, they only looked at this particular group. And here you can see at these people 18 to 65 years not at risk, and then these particular risk groups compared to the people of older age. And here you actually see that those people who have severe risk have almost similar vaccine efficacy with the Moderna vaccine, which is really, really hopeful. If you look at the safety and immunogenicity of this vaccine in older adults, there's a small trial, it's open label, only 40 patients was actually added to the original trial with people with age of 56 to 70 years, or really older people with age over 71. And what you can see here is these 2 different vaccination doses, 1 and 2. And you see that the side effects increase with the second dose. And here you see the age groups with the higher age group at the bottom. And actually they're very similar or generally the same side effects. Only for pain at the injection site, you see that people who are older more often complain about that. But in general, actually this vaccine, also in these really higher age groups above 70, is well tolerated. If we look at the immunological response of this subgroup of 40 patients, they were actually quite similar to the responses among younger patients. Here you see the antibody response to the RBD. Here you see the neutralization assays, and here you see the T cell responses. And in general, here you see the people with 18 to 55, And here you see the older age groups. And this is similar for all the graphics. So the older age groups, and here the comparator group with the younger ones. And actually, in general, the antibody response and the neutralizing antibody response are pretty similar among age groups. And we only see with the T cell response a little bit of dampened response in the older population. If we then go to the AstraZeneca vaccine, and this is interesting to know, it's a simian adenovirus vector-based vaccine. So in the general population, we do not have already antibodies to this vector, which is generally a good sign. This trial was done in different countries with different codes, and only 12% had an age above 55 years, and the mean age is pretty young. For instance, the South African part of the trial, the mean age was only 31. In participants who received 2 standard doses of this vaccine, efficacy was generally lower than seen in other trials, 62%. But in participants in the trial that took place in the UK, we had a group that first had a low dose, followed by a standard dose of the vaccine. And there we saw an impressive improved efficacy of 90%. So in this trial, there's a really different situation with different dosing in different countries. Also in South Africa, of course, there's a larger spread of the South African variant that seems to be an immune escape variant. So in this trial, there are actually no age or immune compromised subgroup analysis. If we look into more detail to this AstraZeneca Oxford vaccine, we see that healthy adults were actually enrolled in an age escalation manner. So people with severe comorbidities or high frailty scores among the elderly were really excluded. So that means that it's not really, at this time, very good translatable to the general population. We also see that low-dose cohort and standard dose group were randomly assigned to receive either the vaccine or a control vaccine in an escalating manner, so that there actually are about more elderly people included. The mean age in the highest age group was here still not that high, 73 only. What was really good news was that here the side effects were less common in the older age groups. Which actually is similar for other trials as well. If we then look particularly at the immunity, then we see that the humoral immunity after a second dose, so these are all the age group with 2 doses, and these are different age groups with only 1 dose. But actually, it doesn't really matter if you look at the neutralizing antibody titers in which age group you are, although confidence intervals widen a little bit for the older age group. If we look at the humoral response to the spike protein, actually we see a pretty similar picture. Ones with only 1 dose have lesser humoral immunity, but all the groups with 2 doses, independent of the age, have a really good response. And the T cell response is a little bit lower for people with a higher age group, as we also saw in the earlier data. But generally, this looks pretty good, also in the elderly. Then there's another vaccine which actually used the adenovirus vector. This is a vaccine from China, and it used a commonly called virus, adenovirus 5. There's quite some in the population to this vector, quite some exposure known. And we have learned from other vaccines that this may be a problem, because if people have already seen this virus that's used as a vector, they may respond less well to the vaccine. This was a double-blind, randomized dose finding, a safety trial with a little small group, only 500 people, randomized over 2 doses and placebo, 1:1:1. And only 30% was over 55 years, so there's only a small group of people really older. What we saw was that already 50% had high and 48 low pre-existing immunity to this vector, which is actually a common cold virus. And also during the trial, there was no SARS-CoV exposure in the country. So that means that efficacy was really not something that could be measured. But looking at the dose finding and the safety trial, at least we could say that people who are older than 55 years and had or had high pre-existing immunity to this factor had relatively low antibody responses. And this was the same for the antibodies to the RBD, but also to the neutralizing antibodies. Although they were still higher compared to placebo. The T-cell response was similar among all age groups. So what I'm now planning to do is actually in this group, another trial, a phase 2B, where people will receive a second dose. But they really are titrating to the levels of the adenovirus antibodies because they saw during the trial a boost of the adenovirus 5 immunity. So they're waiting for the best time based on titration to give the second dose. So at this time point, it doesn't look that particularly interesting, but it may, with the second dose over time, improve. Then there's another vaccine that actually uses adenovirus, but this is an adenovirus 26, which has really low seroprevalence. And this is actually the Janssen vaccine. And they performed a placebo-controlled phase 1/2A, randomized for healthy adults between the ages of 18 and 55, but also a specific second or third cohort with people over 65 years of age, which is really nice. This is a much larger group of older people. And they had a pretty difficult trial design with low dose or high dose in a single and 2 dose schedule. But it means a lot of different groups, but it also gives a lot of information. If we first look at the grade 2 and grade 3 events in patients over 65 years, we really see that this is the cohort of the younger people. This is cohort 3, the people over 65. You see clearly a trend to less side effects after vaccination in the older group, pretty similar as we have seen for other trials. If we then look at the humoral immunity, the T-cell response, we actually see a very strong humoral response, both for the antibodies to the spike protein, as well to as the neutralizing antibody, very comparable in cohort 3 compared to cohort 1. And actually a similar picture for at least the T1 T-cell response. So this vaccine looks pretty good in immunity in the older population. They also had the Assemble trial, phase 3, over 40,000 people, double blind, 34% of people over 60 years, which was not in the, this data was only released in a press release. And what we there saw is that 60% effective vaccine in preventing moderate to severe COVID disease, 28 days following vaccination. And there's a pretty big difference between South Africa and the USA, which may have to do with population efforts, but may also have to do with the South African variant at that time was already highly spread in South Africa. Despite its lower protection in South African region, we saw 85% effective protection against severe disease, which is really, really looking good. Also, if we look at the trial, there was quite a big group, 41%, who had comorbidities associated with increased risk for progression to severe COVID, which was, for instance, obesity, type 2 diabetes, hypertension, but also people living with HIV. So this trial were not only healthy people, but really more people from real life situations. So in conclusion, there's a very limited amount of data on SARS-CoV-2 vaccination and people living with HIV, or people with other underlying immunological conditions. But importantly, those people have been included in some of the trials, and we see no safety alerts. That really gives us a good feeling about including of these groups in the trials, but also an advice for all of us to watch those patients and follow them up more actively. If we look in the elderly, vaccines tend to result in less adverse events, especially in this group just over 65. Maybe we don't know, we cannot really translate it to the really old group, like the 90 years old, and that is something we really have to learn from the actual vaccination outside the trial. Generally, the vaccines produce an adequate humoral response and also a T-cell response, which is pretty good. But it's still relating to a somewhat lower overall vaccine efficacy, as we know from other diseases. So I think, in general, we can conclude these data confirm that there is an adequate response of all these vaccines in the elderly groups, except for maybe the adenovirus vaccine from China. And that it's a very good choice that we really prioritize the elderly groups because they do benefit from the vaccinations. So thank you so much. This is what I wanted to share with you and hope to see you in real life soon. Bye-bye.
Well, thank you so much, Dr. Wensing, for that terrific review of immunogenicity and efficacy data in important subgroups. Let me invite all of our speakers and my co-moderator, Anton Pozniak, to all join in now for the question and answer period. We've had really a terrific list of questions accumulating throughout the talks. It'll be a challenge to get through them all, but we'll try our best. I'm going to start off by going back to some of the basics here, and then we'll get into the more clinically oriented questions. So one of the earliest questions we had, which is really reminds us that these mRNA vaccines are actually quite new, is what do we know about the intracellular persistence of the messenger RNA after its delivery, and whether that might or might not relate to any longer term consequences such as autoimmune disease and the like? And I think I'll direct this to Dr. Ogbuagu.
Okay, so for some of the vaccine platforms, like the Pfizer vaccine, for example, we don't have specific data on the degradation of the vaccine, the mRNA. However, we know that the mRNAs are degraded by RNases, and I've seen reports in the range of 24 to 48 hours on average. There's also a different half-life for breakdown of the lipid nanoparticles that are used to encase the mRNA. There's more robust data on that with the various vaccines. So, it's degraded pretty rapidly. And I think part of the advances in the technology was finding a way to protect the mRNA to allow it to be delivered to cells and generate immune response before it's degraded. Great,
thanks. And then another question for you, and this is again sort of back to some of the basics of the clinical trials design. Can you just remind our listeners how was the efficacy endpoint defined in these trials? Are we talking about prevention of infection, prevention of severe disease, prevention of symptomatic disease?
Yeah, that's a great question. So I think we really need to pay attention to those because the primary endpoints for the different trials were a little bit different. So for example, for the Pfizer and Moderna studies, it looked at everyone who had symptomatic COVID-19 disease without prejudice to their disease severity. And some of the more recent reports we've heard are looking at moderate to severe disease. And that's why I thought to show that model in my presentation, showing that it appears that there's almost a increased gradient of protection or efficacy against the more severe forms of disease and death. So that's how it's done. So obviously, because the endpoints have primarily been symptomatic disease, which really is clinically meaningful, right? They haven't really always looked actively for asymptomatic infection, but at least 2 studies have reported those, the Moderna and the AstraZeneca study, and miraculously, both of those have the exact same point estimate of 67% reduction in asymptomatic infection. So we'll learn so much more about that.
Great, thanks. That helps because it gets to one of the other questions that we had as well about prevention of transmission. Dr. Eberhardt, can you remind our listeners about what is the difference between just measuring SARS-CoV-2 antibody, IgG, and actually measuring neutralizing antibody? And does one really correlate with the other, or if you have a spike protein antibody, does that necessarily mean you have neutralizing?
Thank you. Very good question. It's different. So the antibodies that are directed against, for example, the spike protein, which is part of the vaccines, or against other components of the virus, that just indicate that there is binding that happens, but it doesn't indicate the functionality of these antibodies. So in order to know if, for example, they can stop the invasion into a cell, which we say it's a neutralizing antibody, right? Because it's neutralizing the virus. There's another test that is necessary, and this is not the conventional S1 ELISA that we are doing, but it's a neutralizing test. And so there's different ways of doing these tests, as I showed, and it's more or less difficult, and more or less biosafety measures are needed to do a true neutralization assay with the SARS-CoV-2 virus.
And then you, as well as our other speakers, pointed out the induction of T-cell responses. So which are more important, or are they both important in terms of protection? Recognizing that we don't yet have a target.
Well, if I would know, I think we would be very much advanced. So both, we believe both are important, right? I mean, every part of the immune system has its role in defending. And so if you have neutralizing antibodies, this will defend the virus to enter, right, into a cell. But then, for example, T cells, they have a very important role in clearing, for example, infected cells, and so in helping us to rapidly clear infection. And what we believe right now, but we don't have evidence, is that all of these parts of the immune response are important in protecting us from the COVID-19 disease. And what we know is, for example, that we have patients that are not able to mount IgG antibody responses, and they still can recover from COVID-19, which indicates that there is more to it than just antibody responses.
So can I jump in there and talk about the clinical correlate of that? I mean, if you have a severe reaction to a vaccine, you know, not so you get flu-like symptoms, are there any evidence that that's going to protect you better and get you better responses, Th1 responses, than people who go, I had the vaccine and nothing happened, I just got a sore arm for an hour? Is there any clinical correlation between what's going on in your arm after the vaccine and the vaccine response?
So right now, as far as I know, for COVID-19, there is no data on that, right? And it would be interesting to see if you have the innate, so the initial inflammatory response, if this high response, for example, would correlate with a higher antibody response or a higher T-cell response, which would be theoretically logical, right? But we don't have evidence yet to say that. And to be happy to say my arm was swollen, so I respond better than you do.
Yeah, because I know in the meningitis vaccine trials, they gave, they did trial paracetamol, and it didn't affect the vaccine efficacy, but it did help the pain and swelling. So maybe but that's working through a different mechanism, I know. So, but it would be, I know it seems like a strange clinical question, but I think it needs answering because I get it asked a lot.
Yeah, I agree. Yeah.
It is an important issue because we certainly with the Pfizer vaccine and to an extent with Moderna, there was much less reactogenicity in older populations. The clinical data would suggest efficacy was relatively similar despite the difference in reactogenicity. So that may be a hint, but it does raise some grounds for concern. I guess the other couple of points to think about are, we know from both the Moderna and Pfizer studies, and I think we're seeing from the other trials, that we begin to see protection after the first vaccination. And in the Pfizer vaccine, there's, as you showed, very little antibody production yet. Yep. And so that suggests there may be alternative mechanisms such as cellular immunity that are initially protective. But we've seen clinically in our patients who've received rituximab or who are unable to mount immune responses, serologic immune responses, prolonged shedding of virus and longer course of disease. So even though you may be able to recover without making IgG, it certainly seems to help.
Definitely.
So we've had a lot of questions about this gap that Dan just alluded to. So I don't know, Onyema, what you think about this 3 weeks or longer. I'm going to come back to you, Christiane, about the gap and the effect on B cells. But for example, I'm getting my second dose after 9 weeks now of the Pfizer, and we've just talked about the antibody levels. I don't know, what reassurance can you give the general public because this, I know you've showed the data and I've seen data before, but what assurance can you give the general public that we may not get severe disease, but we may get moderate disease, and that may reflect that we could transmit it more? I don't know about that, but I really would just like your views about transmission if you delay the second dose, about whether or not we'll get moderate disease rather than severe, and we would have got minimal disease if we'd have had it on time. Are there any thoughts that go through your head when I ask you those difficult questions? And I'm asking them because I'm sure all of you get asked them all the time.
Yeah. So first point I always like to make is that we're thinking and talking about these because of vaccine shortages, right? If we didn't have a vaccine shortage, we'd all keep fidelity to whatever was studied and wait till we had more data from these extended interval studies to make decisions. So I just want to make that clear. So it's not a luxury for now because we just have the vaccine shortages. And part of the good news, though, is that even after the first dose of vaccine, we're seeing rates of protection. What I always said was we expected vaccines to be 50% effective. And now we're seeing that even after first dose, almost everyone is above 50% anyway, that bar that we set. So frankly, the first doses provide quite robust protection. And so it makes it a little less worrisome about administering the second dose. But I think the second dose does contribute to, like we've seen from immunogenicity data, higher antibody levels. And I think that should also have an impact on the duration of protection. The data that we have about these intervals comes from the AstraZeneca trial, which we showed, right? And it's actually fascinating that there was a clear relationship between the gap or the extended duration and stepwise with effectiveness, which is fascinating. Viral vector vaccines are unique. I think anti-vector immunity may play some role in what happens in between doses. At least that was one of the postulated mechanisms for why the half dose, full dose tended to have higher efficacy, but I don't know that those same considerations apply to other vaccine platforms. And I think we just have to do studies. I think there's some suggestion that if you vaccinate people too closely, you can have almost somewhat of an immune exhaustion. And so there might be some argument that delaying vaccines could lead to more robust responses. But we'd like to see more data to make any final thoughts on that.
And the B cell response, Christiane?
Yeah, I agree that I think we don't know enough yet. The other theory is about delaying, that is that your B cell response after one dose has antibodies that have a quality that is not as good as if you boost it again, and they can maturate more and more. And that, for example, we say the affinity, so the capability to bind, right, that it's less good after one dose. And then you might think that you would need the second dose to have more performing antibodies. How long has to be the interval between both doses? Nobody knows, right? I mean, we all don't know. And we do what we can do. So some countries stick to the 2 doses and vaccinate fewer people, and other countries say, okay, I delay and I try to vaccinate as many, at least with 1 dose. We will see in some months when we have effectiveness data from different countries with different attitudes about what works and what works better. But I think it's a very difficult question.
Annemarie, can I just bring you in here, because the data came from delaying, as you've said, that from the AstraZeneca vaccine. Yet when you show us the data, we don't have that much data on the elderly whose immune responses, as we've seen from this, isn't as good. So what's your take on the delaying vaccines?
Yeah. Well, I think it's also, we have to be careful because the trials have different endpoints, or actually they did analysis interim. So some of them, people were really quickly after a dose, and others people were longer away. So then I think that also, a little bit blurs the protection rate from the efficacy data. I think everybody was quite surprised, I think, from the first data from Oxford that the AstraZeneca vaccine, that you had actually a better response if you waited a little bit longer. I would not have thought that up front. I think Christiane can maybe better explain than me, because I was really surprised about it. And I also tried people that it may not be translatable to other vaccines, so that we just don't know that much about it. I don't know, Christiane, what do you think? What would be here the main driver? But for me, it was quite surprising, actually.
Yeah, I think one thing we should be careful perhaps about is that the trial was not made to investigate the duration between doses, right? And this is a post-hoc analysis of kind of, let's say, an accident that happened where the first dose was given just half dose, and then time went by until it was decided how to continue, right? And so I think it's very interesting, and I think it's good data, but I think it's not solid enough to base everything on it. I think having 2 doses is kind of the double prime, what we do with other vaccines, with protein-based vaccines, where we give 2 doses at 1 month interval, and then after 6 months, the booster dose to induce long-term memory. So this could be something to explain, or to say, well, we would need to give 2 doses at a very short interval. But then we say it's an RNA vaccine, so we don't really know if this is more like a vaccine, like a live attenuated vaccine, where we might not need this, right? So yeah, it's a very good question, and it's open answers.
Annemarie, what can we say about the safety of the vaccine in people who want to become pregnant, or in people who are pregnant?
Well, I think, we don't have a particular reason to be worried about people who are pregnant. But of course, with any drug used in pregnancy, you would like to avoid. And it's, I think, the independent risk for an individual to see, somebody who may have underlying conditions and be at severe risk for a COVID pattern that maybe has a dramatic, evolvement. You want to vaccinate somebody who's pretty young and pregnant, you may want to say, wait until you have delivered your baby, and, listen to all the rules that you have to take into account to avoid becoming infected. So I think at this time point, we don't have a lot of data. And I think the individual, situation of the pregnant woman should actually advise you to vaccinate or not. But I don't think we have to be too worried, especially, if you look at how the vaccines have been done. I mean, we have quite some experience with the adeno from Janssen. And I also really don't mechanistically could think of why the mRNA vaccine should be more of a risk in pregnancy. I don't really see a reason that we have to be very concerned.
What about...
Can I add a...
Okay. I'm sorry, go ahead, Onyema.
Yeah. So I just wanted to say that this is an important issue because, in the United States, for example, 76% of our healthcare workforce are women. And so this is just certainly, and remember, healthcare workers are uniquely at risk by virtue of our jobs. And so it's a big deal for, considering the risk and benefits for pregnancy. And now that there's at least, evidence that pregnant women can have worse outcomes with COVID-19, it tilts the risk benefit in spite of the unknowns to having pregnant women do that. And just to say, I don't have the most updated numbers, but I know close to 20,000 pregnant women in the US, if not more by now, have received the vaccine. And there are pregnancy registries to capture some of those outcomes. And both in the Pfizer and Moderna trial, we had people get pregnant all through the spectrum of the trial, in between doses, after doses, etc. And to date, we haven't really heard about any concerning safety events, although the pregnancies are not completed, but nothing worrisome yet.
Christiane, can I just further, are there any vaccine studies where pregnant women who have a non-live vaccine don't respond as well? Are there any vaccines that happens in?
I think so. Most of the trials, for example, influenza, this is one of the vaccines we like to give to pregnant women to protect them and then afterwards the offspring. There is trials that have been made, but often they were not directly, the control group was not pregnant women to really assess it, but it was different groups that were tested in trials. We know that they can respond. Yes, there is a difference sometimes in immune responses. And for example, pregnant women are more at risk for some diseases in the last trimester for some viral diseases. But in general, the impression we have for the vaccines that we give during pregnancy, which are pertussis, for example, and influenza, we think that they still have mounted good response and at least a sufficient response. So I don't think that we should need to be worried about this aspect, but more think about the higher incidence of severe disease or even death in pregnant women, right? And then to assess and vaccinate them if they want to.
And Onyema, we're probably going to get data on women who become pregnant while they're during the protocol of being vaccinated, aren't we? Because they're really, because we're all anxious about, say, giving drugs when you become pregnant, and giving vaccine when you become pregnant might be also important. So do you know if people are collecting this data?
Yeah, so we had, yeah, great. So you're right? Because, especially during embryogenesis, the very early parts of pregnancy, that's where you are probably most concerned in the spectrum of pregnancy. We had 23 in Pfizer. I think there were about 13 in the Moderna study. And obviously those patients are being followed over time. And so, obviously you're looking at pregnancy outcomes maybe many months down the road. The studies really just started enrolling in July. So still a couple of months before we get that data. And I think there are pregnancy registries with the vaccine rollout as well. That, that's passive reporting. It's not active surveillance, frankly. So it's not, it's not the perfect system, but I think it's the next best thing with the vaccine rollout.
And also, if I can add some, another point is if you look into pregnancies or into vaccination with live attenuated vaccines in women who become pregnant and who don't know that they are pregnant, there is no higher risk for any problem for mother or fetus or even in the embryogenesis. So if live attenuated vaccines, although we don't give them during pregnancy, are not provoking anything, then as Annemarie said, the mechanism of mRNA is very theoretically impossible that they would be worse than that. So
yeah, but important to collect the data.
Absolutely, not only on pregnant women.
No.
Yes.
Annemarie, what about people with uncontrolled HIV infection? What can we say about efficacy? And I think the Novavax trial particularly might be informative.
Sorry, I didn't hear you quite good. Did you say uncontrolled HIV?
Yes. What can we expect in terms of efficacy if the vaccine is given to people with uncontrolled HIV? Are there any data, especially from African populations, high prevalence?
I haven't seen any data. I know there are some edited people, but most of them are well controlled in the clinical trials, but they're not specifically analyzed at this moment. I think there is a general, people are cautious if people are not well controlled, but we have to realize, I don't think it's really about control of the virus. It's more about states of the immune system. Because if somebody has, really good CD4, but the virus is not completely controlled, then I think you still have a good chance that actually a lot of chance that your vaccine will work. So I think there is the most, a question. If you have a really low CD4 count, how well will you build up immunity? There's also some people who have been afraid of integration of the mRNA vaccine if they have active reverse transcriptase in their bodies, but I don't think that's something we should worry about because we don't have the primers to put this vaccine in our genes. So I think that's also something that will be good just to message and to tell people about.
Onyema, do you want to?
I might. Exactly. Yes. So Novavax the data for Novavax is a little concerning because I think for the subset of people with HIV, actually the vaccine did not show efficacy, in fact, negative in some cases. But again, the uniqueness around HIV in South Africa, obviously, is the circulating variant that was present at the time. And I think they're relatively, but it was quite concerning, and I am holding my breath to see what the data was. And these HIV patients were actually cherry-picked, right? So these people were on therapy, had robust CD4 counts. Now, in HIV medicine in general, we tend to delay vaccination, especially for people whose CD4 count is less than 200 because of suboptimal immune responses. But in this case, these were people who actually met that criteria. So I think there's a lot more to be learned as to, there may be other confounders there, but I think that's the data in HIV patients in a part of the world that has a high prevalence, and so we'll need to see how that plays out.
So can I pin you down there a little? If you had to vaccinate an HIV patient with a good CD4, which one are you going to choose? Which vaccine?
I would go. So first of all, if there were live attenuated vaccines, I think that's the one we'd all be worried about, especially those who had a lower CD4 count. We'd like to make sure it's safe. But if I had a choice, obviously I would go for vaccines that had the much higher efficacy, frankly, in those individuals, especially given the expectation that there's at least some signal for the protein-based vaccine that they tended to have less protection. But again, those numbers were small, and I don't want to conclude on that.
No. It's just an interesting question.
Yeah, the Novavax data are really quite concerning, because I think, if I'm remembering the numbers correctly, overall, when they excluded people with HIV from the analysis, the efficacy was about 60% or so. Yep. When they added them in, it went down to about 49%, and there was only a small group that were HIV infected, maybe 15 or 20%. So that suggested basically no effect in order to drag down the mean efficacy. So that's
and those people without HIV, were they from the same geographical area?
It's all in South Africa.
Oh, in South Africa. Okay, I haven't seen that. Well, I think, it's really puzzling. Also, I have heard a lot of stories from colleagues in South Africa at the moment of people with very clear, serious reinfection. So, have been admitted with COVID, completely healed, and then now with the immune escape variant again, going to IC wards. So I think that's something, we have rarely seen, at least in our setting. So I think, this variant maybe even, have more of an issue if you have HIV, may respond a little bit less. And then you have this variant that is an immune escape variant, and maybe that together makes people with HIV in a more worse position. I don't know.
I think that most of the infections in the Novavax file in South Africa were the new variant. They had a substantial number of infections. Let's switch gears a little bit here. What about vaccinating people who have had, should they get the vaccine? When should they get it? And since they already have some induction of immunity, would a single dose be sufficient if you're looking at a vaccine that would otherwise require 2 doses?
Yeah, was that for me then?
Anybody, but go ahead.
Okay.
All of you have to answer that one.
Okay, fine. I'm happy to start since I started talking. So in the trials, we excluded people with active disease, both by swabs and just symptom screening coming into the study. But I think in both Pfizer and Moderna, I think Pfizer was 3%, maybe Moderna around 2%-ish of people who came in antibody positive. So they probably had asymptomatic infection. Degree of protection was pretty similar. Now, that said, we do know that people who recover from disease, particularly those who have symptomatic disease, and particularly those who have more severe forms of disease, tend to have much more, at least whatever antibody levels mean, tends to be more robust and last decay slower than those with milder form of disease. And clinically, based on our observations, it doesn't appear that people seem to be protected for quite a few months, right, after their initial infection. But there's at least data now, I think, from the Pfizer vaccine that people who have recovered from COVID in the past, when they get a single dose, it's a preprint, so it's not peer-reviewed or anything, but at least no less interesting, that people who have had prior infection who get a single dose of the BNT162, which is a Pfizer vaccine, have antibody levels that even exceed those of people who've gotten 2 doses of vaccine by quite some fold, frankly. So, and I'm one of those of the opinion that probably those who recovered from COVID, especially those who are recent recoveries. Based on this data, again, we need to be cautious because it's not published or peer-reviewed yet. It seems to suggest that a single dose acts to booster, probably boosting that the memory of the prior infection, and that should be sufficient. And also because they feel miserable. The side effects that a lot of these people have are accentuated, and we've all had to be called on people who had COVID before, got the vaccine, and felt awful, and were even so scared that they didn't want to get a second vaccine. And I think now they have an excuse with this data to point in your face that, hey, it looks like I'm even better than the person who was uninfected who got 2 doses. That's what we know for now.
Christiane, what about data that's come out of other studies, say, where people have had the disease and then get vaccinated? I mean, with rabies, I suppose you have it at the same time, so it's not quite the same. But there must be with chickenpox vaccines.
Chickenpox vaccine, the problem is it's a live attenuated vaccine. And so if we see this with Zostavax, which is a, I think, 14-fold higher dose than the initial chickenpox vaccine to boost against shingles, and it's not working very well because it's a live attenuated vaccine. So we don't know for that. But in general, I mean, we know that if you give a booster, every booster normally, or most of the time, improves your response. So it's logical that if you had the disease and you get a booster that after the first dose you have a way higher immunity than somebody who gets the first shot, which is reassuring and good. I just want to come back briefly to the point that Annemarie made about the reinfection of infected people in South Africa with the new variant, right? Which kind of indicates that although we know that immunity or antibodies last we have no data for 6 months and even longer, right? But that those people, when they get infected with the mutant, it indicates that probably we need a higher response or like a more neutralizing antibody or whatever that is. And this is worrisome in this theory about delaying 2 doses, right? If we are in an area of mutants, and then we might not have an optimal response. Yes, against the classical strain, but not against the mutant between the 2 doses. So I think this is something that we should be very careful about. And
But I suppose if you had a vaccination and then got the mutant afterwards, we haven't seen the data that mutant will cause severe disease enough to put you into hospital. And then you had a second dose, you would be you might be boosting an immune response that you got to the mutant, though
this I think, and you
more
might
specifically.
You might either you can broaden your spectrum, or you have this original antigenic sin thing where you just respond exactly to the same what you have seen first. But I mean, this is the 2 theories. We are going to see what's going to happen. But I think that if people who had the infection and they get reinfected and they're also admitted to the ICU in South Africa, we might probably not be very well off with just 1 vaccine dose.
No.
Right?
Okay.
Annemarie, quickly, because we're going to run out of time. What about other immunocompromising conditions? People on immunosuppressive therapy, people with hematologic malignancies. What do we know about the use of vaccines in those populations?
Well, I looked into the trials, and I don't think there's a lot of exclusion of those people. So I think only one of the trials really had a larger group of people who had immunocompromised conditions, but they were not listed what they were. And I think it's such a big family of things, so we really have to be careful to make one recommendation. And I think for HIV, we're pretty sure that people will mostly benefit from vaccination. I think it has to be one by one, considering what kind of disease people have, and ask their caring physician what they would advise. And of course, we have to be careful with live attenuated, but I think most people will generally benefit from vaccination.
What happens in
ongoing studies trying to look at populations?
I'm just concerned about people on very high dose immunosuppressants or on high dose steroids, whether they'll have is there any data from anything that we've been doing? I mean, even in previous vaccine studies, that's not COVID, that they respond or don't respond.
Well, that brings us to the end of our time. I want to thank everybody. It's really a terrific roundtable. We also had 3 terrific talks. So thank you, Onyema, Christiane, and Annemarie. There's going to be a 15-minute break, and then we'll continue with the second session, and Francois Venter and Katie, blank on her name for a second. Stevenson, my colleague from across the city, will be the co-moderators, and I'm sure it will be just as interesting a session as this one. So thank you all very much.